name is Françoise Barré-Sinoussi, and I am the co-chair of the IS initiative towards an HIV cure. I like to say that it's a pleasure to co-chair this initiative together with my colleague Sharon Lewin and Steve Dix since now several years. Before we start, I'd like to make an announcement. It will be an action organized by the international community of women living with HIV at 10:30 a.m. at the main entrance of the Vancouver Convention Center. This action is really for women in die-in to demand indeed an end to institutional violence in healthcare setting and structural violence experienced by women living with HIV, and also to remember people who have died from complications of HIV. Once again, it has been a fascinating few days regarding HIV cure. We held a very successful HIV symposium over the weekend. It has been our biggest ever turnout with over than 300 participants this year. As usual too, there has been a lot of interest from the media. Many were in attendance over the weekend, and as you know, many more have covered the case of the French teenager in remission, which Asier will talk to firstly, and no doubt you will also have questions about this issue. I wanted here to make some very brief introductory remarks now. It's very clear that in the past 2 years, the field of HIV cure is moving very quickly. The viral rebound of both the Mississippi baby and the Boston patients, while of course disappointing, must also be considered, be viewed as a part, let's say, of the learning process in science. The French teenager case is not so different. We will undoubtedly it will undoubtedly teach us new things. We need certainly to develop better tools to measure the virus while people are on treatment and once they stop treatment, including the need for better tools and biomarkers to evaluate or predict the response of therapeutic strategy. We need to have a much better assay to really know where the virus also is hiding in the different compartments of the body. Not only do we need to tackle the virus that persists on treatment, but also we need a good immune response to control the virus that may emerge. Early treatment. Early treatment is certainly part of the science regarding HIV cure, but early treatment is also a priority for healthcare way forward for people living with HIV. We know already that the successful maintenance of viral load over the past decade by the Visconti patients in France was probably due in large measure to the fact that the treatment was in fact started at a very early stage. Industry is becoming more and more engaged in funding, which is vital if we have to move forward. With that, I would like to hand over to my colleagues. I will first introduce our first five speakers, 4 or 5, because we are waiting for Sarah Findler. They will briefly talk about what they presented here at the symposium. I will then turn to my co-chair, Steve, and Sharon. Steve will moderate the discussion with you. And Sharon will provide some conclusion and a reflection of where we might be moving over the next, let's say, 12, 24 months until the next conference in Durban. I will like to introduce the speaker. First, John Mascola. John is the director of the Vaccine Research Center at the NIH. John will speak briefly about the potential role of broadly neutralizing antibody, I guess, in HIV cure approaches. Sarah was supposed to speak then, but we will see if she arrives. Asier will be the next speaker. Asier is the former collaborator from my own laboratory at Pasteru Institute. He's assistant professor now in the new unit of Michaela Müller-Trudvin at the Pasteru Institute in Paris. Asier will talk about the 12 years virological remission of the French teenager girl after early initiated antiretroviral therapy. Christopher Peterson. He is a postdoctoral research fellowship at the Friedrich Cancer Research Center here in the United States. Chris will talk about a fascinating study indeed on the zinc finger nuclease gene editing for functional cure in a non-human primate model. Lynda D is an HIV/AIDS treatment activist, and she was a co-founder of AIDS Action Baltimore in 1987. She's currently a member of the Community Advisory Board of CARE, the collaboratory of AIDS research for eradication. Lynda will talk to issue around the future of research funding and its impact on community. So, John, would you like to start?
So thank you, Francois, and good morning, everybody. I'm going to say a few words about some of the discussion we had at the symposium with regard to the role of antibodies as part of the cure agenda. Over the past 5 or 10 years, the field of antibody research has come to understand a couple of important things. One is that some HIV-infected individuals make remarkably potent antibodies against the virus, and researchers have been able to isolate those antibodies. So we have them in hand as potential clinical products now, and they can potentially be used either for prevention of infection or treatment of infection. So what we talked about with regard to treatment are that there are several areas where one might consider using antibodies. First of all, the recognition that antiretroviral drugs remain the cornerstone of treatment. So the question is, where could antibodies potentially play a role? And there were several that we talked about that I mentioned in my talk. One was that antibodies may play a role if given very early in infection with antiretroviral drugs, to bring the virus under control very quickly and therefore have some impact on the latent reservoir. The other area would be to help maintain viral suppression. So once individuals are well suppressed on oral antiretroviral drugs, the question is how long can they be maintained, and is it possible to either withdraw antiretrovirals or move to a very limited regimen. And here there may be a role for antibody to help maintain viral suppression if antibodies can be made to have a long circulating half-life. And lastly, and potentially most importantly, would antibodies have some effect that antiretroviral drugs don't have? And that in one area that may be possible is the ability to kill HIV-infected cells. So antibodies have a well-known effector function that allows them to bring in close proximity certain types of immune cells that can kill virally infected cells. So if antibodies can mediate that kind of cell killing, they have a potential, if given with other types of treatment to, for example, induce expression of virus from cells, they may be able to kill cells and affect the viral reservoir. I'll stop there.
So, good morning. What we have described during the conference is the case of a young girl in France who was infected during the late pregnancy or delivery and received an antiretroviral treatment very early. First a prophylactic treatment initiated the day she was born, and then at 3 months of age a combinatory antiretroviral treatment that was kept for probably 6 years. This antiretroviral treatment was discontinued during a loss of follow-up of the family. And 1 year after this loss of follow-up, when the family came back to the clinical center, the viral load was observed to be undetectable, despite the fact that there was no antiretroviral molecules having been given to this girl for a few months. So this girl had stayed without taking antiretroviral treatment for 12 years now. What we know is that this girl is still infected. We can detect HIV DNA in the cells of this girl. But viral replication in the plasma is undetectable. Currently, even when we use very sensitive techniques in the laboratory, we cannot detect the viral replication. But as I say, we can detect the HIV DNA in the cells. Clearly, the virus is still there, and actually we can reactivate some viral replication when we isolate cells and we activate them in the laboratory. We don't find the factors that are commonly associated with natural control of infection, and so what we are trying to do is try to better understand which are the mechanisms associated to this remission of HIV infection. But what seems to be associated, at least partially, is the fact that the treatment was initiated very early and that the treatment was kept for several years, because during her early life, this girl was not able to control infection when the treatment was not efficient. And obviously, we believe that there are a combination of factors that are contributing to this remission, because this case is quite exceptional. It's something also that we want to make very clear, and that most HIV-infected patients, either children or adults, that even if they start the treatment very early and they stop this treatment, they will lose control of infection. This is something that we have also learned looking at the data from the HERONEWS score.
Thank you, Asier. Chris?
Thank you. I'm here to sort of represent the gene therapy angle for HIV cure. I work in Hans-Peter Team's lab at the Fred Hutchinson Cancer Research Center in Seattle, and we have a very broad interest in gene therapy for a number of human diseases. We're involved in clinical trials of gene therapy for cancer, brain cancer, Fanconi anemia, and HIV. The work that I talked about this weekend, and I'll also be talking about today, has to do with using a preclinical model that utilizes non-human primates to conduct stem cell transplants. So we're taking stem cells out of these animals, and we're engineering those stem cells with a gene that's involved in the ability of HIV to get into cells. So by engineering these stem cells and then putting them back into the animals, those stem cells can then give rise to all the different sorts of blood cells, many of which are susceptible to infection. What we've been able to show is that our ability to engineer these cells and safely transplant them into our preclinical model is effective, and we can see basically the blood system with these cells, so that we have protected T cells that shouldn't be able to be infected, and what we're starting to do is look from there. The two ways that we're thinking about how this might be beneficial are, number one, if we put in enough of those protected cells, the virus shouldn't be able to spread, and we would be able to look at functional cure. The way that we're really focused on right now is that even if just a proportion of those cells are protected, it's really able to boost the immune system. And if only thinking of these different cells as maybe better protected soldiers, if we have just a few of those soldiers that are better able to respond to the virus, that has a beneficial effect on the entire immune system and on the entire blood system. So we're in the process right now of working with our industry partners. We do a lot of our work in collaboration with Sangamo Biosciences out of California. Sangamo has already shown that the methods that we're using to edit stem cells also work if you want to edit T cells. So we've enjoyed a great collaboration with them, and we're moving forward to learn more both in our preclinical model, and Sangamo also has an early phase clinical trial that's going to be starting with the same technique very soon. So we're interested in looking into the future of ways that we can increase the number of these protected cells in our animals, as well as in the patients in the clinical trial, and just getting a better idea of what the correlates are of protection that our gene therapy strategy mediates.
Thank you, Chris, and I must add that the nice work of Chris was awarded the HERONEWS HIV Cure Prize. Lynda?
Thank you. I'd like to turn your attention to a report that the IAS handed out to people that were at the CURE conference that preceded the regular larger conference here. And it's about funding, and it's about CURE funding and how much different nations have actually contributed to that. I found it very interesting. And, you know, although this is an international conference, I'm from the United States, and we can see from this graph, which I can certainly share with you later, that the United States significantly contributes a higher amount of funding to cure research and AIDS research, period, as well. And I'm talking about this because we could really use your help, because that funding is under attack in the United States. And it's very unfortunate that we're at a time where there are so many scientific opportunities, and we've spent so much money and time and effort on HIV and cure research. It would be very sad for it to be cut off at this point. So we could really use your help with that. And the other two things that I'd like to say just really quickly, you know, it's very important to report work that is exciting. This 11-year-old child has been described here. But it's also very important, we feel, to keep expectations reasonable for legislators and for the community, so that people don't think, well, there is a cure. Because oftentimes, I mean, we have more questions than answers in many of these cases, and people have a tendency to look at the top line sorts of, okay, well, AIDS is over now. You know, many people in the United States, for instance, believe there's a vaccine. So I think it's incumbent on the press to be very careful about how these things are reported, and also to understand that even if there's not a cure, functional, complete, that there are oftentimes, you know, the work that's being done, while it's not a home run, leads to first base and second base hits, where people actually, people with HIV actually will have better quality of life and better morbidity and mortality as a result of information that we find out in these trials along the way.
Thank you, Lynda. I just add before handing over to Steve that it's not only in the United States. We can have the same situation in Europe, maybe Sharon can say for Australia, but I think we are all to be really worried for the future.
Okay, so we're going to now open it up for questions. Let me just make a few comments just from my perspective, where we are as a field. You know, there's still a tremendous amount of need for more basic investment into basic discovery, and that's ongoing. I'm encouraged that actually a lot of smaller biotech companies are getting involved in cure research and willing to make some high-risk bets in this arena. It should be noted that the cure of hepatitis C, actually, this was commented at the meeting, hepatitis C cure came from the biotech industry. And so that's encouraging. But the field's maturing, as it needs to do, and we are now actually in a transition, I believe, from where we're going from hypothesis-generating studies like the teenager to formal, prospective, careful investigation in phase 1, phase 2 type studies, and a few of them are being launched, and we can discuss that if that's of interest. But I'll go ahead and, Red, you have a question?
Yes, my name is Rich Shaik. I'm with Ephora TV, and I'm asking Christopher the question that the genetic approach that you're using, is that sort of a dual immune system, or is it some sort of enhanced immune system? And if so, if it's dual, how does it not compete for dominance or something like that?
So we're using a nuclease platform, and there are a number of maybe broadly better the term is gene editing platforms, that we're using proteins to clip out a gene of interest. In our case, we're clipping out a co-receptor gene called CCR5, and we know a lot, the field knows a lot about CCR5 based on the case of the one person that's been cured of HIV named Timothy Brown, who was transplanted with cells that were mutated at that gene. So we know that people can live healthy and full lives with that mutation, so it's not a matter of augmenting the immune system per se. I think that the data that we have so far in engineering these cells suggests that there's no augmentation that we can really pick up other than the fact that the virus can't get into those cells.
Yeah, thank you. Michael Smith, basically, my question is also for you, Peters. Just to, actually, I have several quick questions, so I think you can answer them probably in 1 or 2 words.
Sure.
I hope so. First question, in non-human primates, you've obviously performed this procedure. Have you tested them against HIV to see if it actually works? Secondly, there's speaking of Sangamo and others, other people have been working on similar approaches, but using T cells as opposed to stem cells. So I'm assuming that's the major difference between the work that's gone on in humans and the work that you're doing. And tell me if I'm right or wrong. And secondly, what do you find in terms of the duration of the cells? Do you have to do like a complete conditioning regimen on the animals in order to put in the engineered cells, or can you put them in and have them engraft and take over the immune system? How does that work?
Yeah, I'll answer the third question first, and remind me if I forget one. But as far as the duration, these cells do engraft long term. We don't see loss. The work that's going on in clinical trials that are coming up that's being run by Sangamo is going to use a lesser conditioning regimen. So the question there is exactly what you just asked, whether the patients can undergo less conditioning and still see those cells engraft long term. The second question about the difference between this approach and the past Sangamo work in T cells, again, the conditioning regimen is one thing. In the early patients, they didn't receive a conditioning regimen, so the T cells could just be put in. There's been more recent work where they've undergone sort of reduced intensity conditioning. I think there's pros and cons to each approach, and we're obviously in our lab interested in the T cell approach and the stem cell approach. And remind me again the first question.
Actually, I think the last question was, have you actually challenged?
Oh yes, so yeah, we're doing it both ways. We have animals where we've transplanted them and then infected them with an HIV-like virus, and we also have animals, which is our preclinical model, where we basically infect them with the virus and suppress them on antiretrovirals, so they're just like a patient, and then we transplant them. We're very early, so we're not prepared to give a lot of data yet for the HIV challenge or the HIV-like challenge, but we're definitely seeing promising results in the very early data.
Deborah Jones, AFP. Several people mentioned funding throughout the conference. I'm wondering if somebody could put some numbers on the existing funding and how much you see at threat, both in Europe and in the United States, please.
This is for HIV research in general or cure research?
Both if you can get it.
We have tried to figure that out.
It's hard to get a grasp
Well,
of
it's
the
pretty hard to figure out the NIH coding. So you could have one thing like monoclonal antibodies that are coded one way, but that really also affect cure research. So, I mean, the way we look at it is NIH funding just generally for all the institutes, period. And we try not to pit diseases against each other, but for the last, God, I don't know how many years, HIV across the board has gotten 10% of the NIH budget, and that is actually what's in jeopardy. You know, this has a tendency to if you want me to tell you the truth, whoever has the best advocates, and at this point, who has the most money to deal with Congress and you know, advertising and all the stuff, you know, television commercials and all the stuff that goes with that. So, you know, this really should be based on scientific opportunity and really grounded in where we are, and not just who has the best screamers, you know, I guess, for lack of a better word. So that 10% is in jeopardy in the United States, if that helps you.
I can just give you the level of funding in France for the National Agency for AIDS Research and Viral Hepatitis. It's around 100 million of US dollars for both HIV and viral hepatitis research.
Is it possible to put a figure on how much is needed and what the gap is? I mean, it's one thing to say we need more funding, which everybody says, but writing for people in other countries, how much?
Billions is the number.
Yeah.
It generally requires billions to develop these types of.
And really, if you want to do it in a way that's reasonable, look at where there are the scientific opportunities. And, don't pour billions into something that doesn't have the infrastructure and the wherewithal, or the science is just not ready for billions and billions. I guess that's sort of the point. So, I mean, the IAS chart here is very good about how much has been put in. And I mean, you can see the lines where the only one that's really gone up is the United States and maybe Australia. Or maybe I'm sorry, I'm putting my glasses on.
Yeah, I think that document is quite interesting from the cure research, and overall it's increasing, absolutely dominated by the US, so that other countries, that high-income countries, should be supporting this. This is going to have huge impacts on how many people we can treat, but yet Europe, Australia, Japan, countries, high-income countries are not contributing to the same level that the US is. And overall, as a proportion of total funding, cure research actually remains, I think it's less than 1% of total HIV funding. Those numbers are in there.
So when you're talking about a concern, you're talking about both AIDS research funding and cure research funding?
Yes.
Yeah.
And both are in jeopardy, you're saying?
Yeah, well, cure comes within that ambit of the whole, HIV portfolio, but I think the priorities are vaccines, prevention, comorbidities, and cure. So, and I'm not going to say who gets more or who should get more, and that's my story, and I'm sticking to it about the best scientific opportunities and the best scientific agendas and that sort of thing, and that's how you should look at it, and that's how it should be based. But unfortunately, things don't work that way in the United States Congress.
Hi, Matt Sharp. I'm a person living with AIDS and AIDS cure research activist. And you probably all know what I'm going to ask you, or talk about. One of them is the importance of gene therapy, and I want to just point out that Saez-Ciron gave an excellent talk yesterday, sort of summarizing the field of gene therapy research. He kept saying over and over, it's not a pie in the sky, it's not a pie in the sky. We have a lot of clinical research going on right now. And one of the key things that I think is being talked about more at this conference, which is exciting, is this immunologic effect of modifying these cells. And I wonder, Chris, if you could talk a little bit more about what further research may be happening into that. And then I have a follow-up question for Steve about information.
Yeah, so I think the concept that almost all of us are aware of within the field is that the cells that are being primarily attacked by the virus, CD4 positive T cells, are the same cells that are responsible for coordinating the immune response against the virus. So to go back to the soldier analogy, it's sort of the best soldiers for this particular foe are the first ones that are falling. So the fact that we can even protect a subset of those is being shown to be really beneficial. And we're using this gene editing strategy that I'm talking about today, but we have a number of other strategies that we're interested in the lab where we can express small genes or small proteins that can block the ability of the virus to get in. And it's very clear from those studies that with just 10% of the cells being able to be protected, we see that the ability of cells that are specifically able to destroy the virus and virus-infected cells, those are able to grow out and function a lot better.
And Steve, Peter gave, your colleague Peter Hunt gave a great talk yesterday about inflammation and the issue with persistent inflammation. Even though someone may become cured or rid of the virus, that they'll always have persistent inflammation problems after that. Can you talk a little bit more about that and what's being done? I mean, you could probably talk all afternoon about it, but just give the audience
there is chronic inflammation in almost anyone with HIV, including at very low levels these controllers, quote-unquote lead controllers, people who lack virus. There's only been one person who's cured, and that person actually had remarkably little chronic inflammation. So if you're a fan of anecdotes, as I am, I actually found that observation fascinating, and it does suggest if we can remove every last virus particle from a person, this state of chronic inflammation and its associated morbidities may go away.
Even in the gut?
Even in the gut. That's what we did. We did a study of his gut mucosa, and it was perfectly normal. It was remarkably normal, the Berlin patient. So we obviously need more than one example, and we're working on that. It's going to be a while. Any other questions?
You mentioned that there are phase 1, phase 2 studies
Yes.
to respectively look at different strategies. Are they being presented at this meeting, and could you elaborate on that?
Well, Sarah Fiedler actually outlined who could not make it here this morning, but she's leading in the United Kingdom one of the first large shock and kill studies, where they're going to take people with early HIV infection on therapy and randomized to get a shock and kill strategy, to try to shock the virus out of its hiding place and give a vaccine to kill it. And it's a randomized, controlled study, well-powered study, and it's not a definitive study. It's not going to lead to an approved drug, but it's prospective and it's exactly what the field needs to do. Based in large part on the work of Asier and the French, there are other initiatives going on to take people who are on long-term therapy and interrupt drugs in a very careful way to see if we can find these so-called post-treatment controllers and find out, is this real? Because it's debated. And if so, figure out who can stop therapy early and the mechanism for the control. So that's where the field is. And there are several other studies. There's a really innovative one going on in Europe looking at antibodies in combination with a shock strategy. These studies are generally on the order of 20, 50, 60 type patients, early phase 2 type studies. But that's where this field needs to be as it matures. And we won't have any results from that. These types of studies trust me, I do these studies for a living. They typically take a year or two to get approved and a year or two to get, so it's going to be a few years before we have any answers.
Well, we're at it. Has anybody got any idea what makes this young girl different, special, able to be off antiretroviral therapy when all of the other patients rebounded after a certain amount of time?
So for this particular girl, we don't know why she's able to do that. As I said, we think it's a combination of factors. One of them being early treatment initiation. But we're quite convinced that there are host factors that are playing a role in helping this girl and also the patients, the adult patients from the Visconti study that we presented a couple of years ago to keep a remission of infection after treatment interruption. In the case of the patients in the Visconti study, we have an indication that perhaps innate immunity may be playing a role in this control of infection, because we have observed that these patients in the Visconti study have particular cells, natural killer cells, with an enhanced capacity to counteract HIV. What we need to know is to better understand whether this is contributing really in vivo to maintaining this remission of infection, or is a consequence of maintaining this control what we are observing. So we need still a lot of basic research to better understand this system.
Hi, I'm Pamela Fairman, Vancouver Sun. John, I wanted to ask you a few questions about antibody research, because it seems to me that the HIV antibody research will inform lots of different fields in medicine. So you mentioned that you have a collection, I don't know if it's a biobank of antibodies. How do you use them? And if you can just sort of describe in a colorful way how you acquire those antibodies, and then what you do with them. When you find out about a patient who's exceptional to you, in a rush to get their antibodies sent to you, or how does that work?
Sure. So, in a sense, we do something like what you're asking for. In order to find the best antibodies that might actually play a role in the cure agenda, we have to screen and select out from the people who are infected those that make really remarkably good antibodies. And that's done by screening their sera. The antibodies are circulating in the sera, and testing those sera to see how well the sera neutralize virus. Then we go back to that donor and ask for a sample of blood cells, B cells making antibodies. We isolate the antibody cells, the B cells from the donor, and genetically we can then recover or amplify their genes that make the antibody, and that allows us to actually make the antibody in the laboratory and eventually make the antibody in the clinic. So in general, the field now has, at the laboratory level, many such antibodies that target different areas of the virus. And as Steve said, the first clinical trials are now starting where antibodies are being given with shock and kill strategies or giving to people who are very well highly suppressed to see if the antibodies can actually kill infected cells. Did I miss any of your questions?
Well, maybe just a subsequent. Can you tell us how AIDS research has in fact informed other fields of medicine? Are there any examples that you can think of where strategies have been copied with good results?
So it goes both ways, if I'm understanding your question correctly, but with regard to where the AIDS research has informed other fields, I would say that the whole area of AIDS immunology, studying B cells and being able to isolate antibodies in a very efficient way from B cells has emerged in large part from the AIDS research lab. And so antibodies are used for lots of other diseases, cancer diseases, inflammatory diseases. But the technologies to efficiently acquire these antibodies has clearly been helped by the AIDS research.
Can I follow up on what John said? So from a gene editing standpoint, the investment that's been made in the type of strategies that we use for HIV are very applicable. All of the data that we're sharing at this meeting I think are really important in a more general sense, because right now we're targeting a gene involved in HIV susceptibility, but the same strategy could be used in targeting a mutated gene involved in progression of cancer, other genetic diseases. And the fact that we're showing that this strategy is feasible can really spread to a number of diseases other than AIDS and HIV.
Can I just make one point about that? Probably the biggest advance that's spilled into other fields is around antiviral development. So the antivirals that we use to treat HIV, highly potent against hepatitis B, so there's now very effective treatments of hepatitis B, identical drugs, and very similar approaches have really led to the discovery of the fantastic hepatitis C drugs that we now have that can cure hepatitis C. That whole area of antiviral drug development spawned out of the HIV drug development.
Hi, Jeff Berry, Positively Aware Magazine. This question is for Asier. In the HIV Cure Symposium presentation that you did, there were 2 instances of poor adherence in the first 500 days, I believe. And could you go into that a little bit more, and could that have possibly elicited some response from the immune system that had a contributing effect towards the remission? And then Debbie Prasad mentioned that this child was very different from the Mississippi baby. I wondered if you could elaborate on that.
So for the first part, just to describe what happened is, so when this girl initiated the antiretroviral treatment, there were 2 episodes in which adherence to the treatment was not perfect, and this was accompanied by increase in the viral loads. So it went up twice to 70,000 and 90,000 copies, quite significant levels, and this had also an impact on the CD4 T-cell counts. So this showing us that during this first period of life, the treatment was not perfectly suppressive. This girl was not able to control by herself. This is something that could impact in experiencing the immune system with the virus, but actually we cannot know whether this has been a critical point or not. It's something that we need to better understand. And we need to look at that also retrospectively. In many other patients, we have several additional examples in which this happened during this time, and whether this had any impact at all on the outcome of the infection. But this significant. In any case, in my opinion, this is already pointed to the fact that while she was not able to control at this very early period of time when the treatment was not fully effective, she was able to do that after several years when this treatment was maintained and in a very effective way. So I think that we will always have this debate as this trial that Steve was talking about will be complete about whether it's the early treatment or are we selecting some people who may have some capacity to control. In this case, at least, well, we can sort of at the very beginning she was not able to control. But is this because this is in a child, the immune system was not mature enough? There's a lot of interrogations. And regarding the comparison with the Mississippi baby, there are differences. There are also things that are quite similar, and we don't know which are the most important things, or the most important factors here. In the case of the Mississippi baby, a combinatory antiretroviral treatment was initiated right away. A very low viremia during the first few days, and then control and keep at that for several months. And then the HIV DNA was barely detectable, except in very rare occasions in this girl, and was lost for follow-up. And then after a couple of years, 2 years and a half, there was a viral rebound. So in our case, we don't observe a viral rebound after many more years, despite that we are detecting very clearly HIV DNA. So I think that this is also interesting and informing for the field, because we are trying to eliminate all the infected cells, and perhaps this is showing us that we don't need to eliminate all the infected cells if we have other means to control the few that may remain. And obviously we need to better understand which are the kind of viruses, where are they exactly, how are they in these cells that we are detecting as being infected. But so yeah. There are differences with the Mississippi baby, but there are also many other things that are similar.
I mean, the Mississippi baby was all about sterilizing pure cure, no virus, with early therapy. And in this case, it's all about finding a way to control the virus. So it's an example of the two types of cure: functional cure, sterilizing cure, and so forth. So we only have a few more minutes, so quick questions, quick answers.
I just wondered if you discussed the Ottawa cohort, which I reported on that from last year's. I believe it was Ottawa. And I think there were 4 children who were exposed to antiretroviral therapy 72 hours after they were delivered. Any update on what happened to them?
I don't think there's any updates here. But that is, again, a very different strategy than what Asier is talking about. That's about preventing long-term infection. And I don't think it succeeded.
There's at least one rebound.
One rebound, right. You know, my take-home message from these cases is that if you start therapy super early, yes, you may prevent infection, but you don't have enough stimulation of the immune system to control what's left. And so this is the balance. And perhaps what Asier has found is a balance where you start early enough that there's not a lot of virus there, but there's enough that you have an immune system that can recognize it. And that's at least a hypothesis.
Could you clarify when you introduced the antiretroviral therapy in this trial?
So first there was a preventive treatment that was initiated the day she was born. This was kept for 6 weeks. The virus was not detectable during this period, and so the prophylactic treatment was interrupted at 6 weeks. And then, because the viremia went up, the combinatory antiretroviral treatment was reinitiated when she was 3 months old.
I just want to talk a little bit about cases. Steve, you said that you've got one case of cure, and this is another case of remission, and the Mississippi baby was a case. What's the value of these cases that we now we're now starting, in case of the Visconti cohort, there's 14, you know, and this child looks very like the Visconti cohort, as I understand it. How do we translate from these sort of events into something that is more general, or is it just a matter of multiplying cases until you can somehow see a pattern?
I mean, that gets into sort of a philosophical question about science and how to approach science. My personal opinion is that these cases are extremely valuable because they raise hypotheses that can be tested prospectively. Asier has a hypothesis that early therapy alters long-term the immune system in a way that actually the virus, you can more or less cure someone. And that now needs to be tested. There's a hypothesis from this work that it involved some unique NK cell activity. This can now be tested. We have drugs that can alter NK cell function. The Berlin patient has this hypothesis that by taking out CCR5, we can cure people and prevent inflammation. And so these, you know, the Mississippi case hypothesis there is that we can initiate therapy in infants and perhaps adults so early that we basically cure people. So all this is raises questions more than answers questions, and those are personally my favorite types of studies because they allow us to design bigger studies, which often fail, but lead to more questions, and that's how science plays out. So hypothesis generation.
Just something, because I think that this is an example of cases. If we go back to the CCL5 thing, so CCL5 delta 32, that is the mutation that was inserted during the stem cell transplantation in Timothy Brown, is the one that is being pursued. Also, ablation of CCL5 in gene therapy. This came from a very initial analysis in 2 individuals who were extremely resistant to HIV infection, and in which this mutation was firstly identified. So there are also an opportunity that if we identify some particular factors that are clearly restricted to that, there may be a translation. And from these 2 cases in which CCL5 delta 32 was firstly identified, there has been molecules targeting CCL5, there has been new approaches. So I think that they might have some values if we are able to identify these factors.
So we have to finish now, but I think we're going to close it with the big picture comments by Dr. Lewin.
Yeah, I've been asked to speak about what we might see in the future in the next 12 or 24 months, and I think there's kind of 4 main areas we'll see advances in. The first is in a number of really important prospective clinical trials that Steve spoke about. One of those is the effects of early treatment and whether people can safely stop treatment and stay in remission. Those studies are happening, including some very interesting studies in Thailand that we'll hear about shortly. Second really important area is about tools to measure the virus and how we all measure different things in different labs, and can we do better to coordinate it. And a very large study funded by the Gates Foundation right at UCSF that's going to look at that. And finally, the area of combination shock and kill. You heard again about that from Steve and John, giving a latency activating agent and boosting the immune system. The second area will be a better understanding of the virus and how to shut it down, maybe silence it or how to activate it. We started with these drugs, HDAC inhibitors, that were pretty crude instruments, and we're learning a lot more that there are better ones to activate, either alone or in combination, maybe manipulating other things like the hormonal environment. The third area is the area of the immune response, and we think both are important, getting rid of the virus and boosting the immune response. And there have been so many major advances in that area in other fields. We've heard about in the vaccine field, the advances in broadly neutralizing antibodies, new vectors. We're hoping that will bring benefits to the cure field. But in the cancer field, all of these new agents that are improving cancer, that block, one example, PD-1 or Keytruda, those immune checkpoint markers or blockers are going to be really important, we think, in boosting immunity in the setting of cure. And finally, there's new ways of working that I think we're going to see and hopefully see the benefits of, new partnerships. We've talked a lot about engaging pharma. I think that is at risk. However, there are some new initiatives, Cura, UNC and GSK partnering I'm hoping we'll see more of those. AMFA putting a considerable amount of money into cure research, establishing a new Cure Institute that will be announced later in the year. And I'm hoping that we can and really see better and more innovative models of working in partnership with pharma, with big pharma, with small pharma, with governments beyond the US government, and more importantly also with the community. I think our messaging around cure is getting a bit clearer. I think we can work a bit better on that. Talking about remission here, not cure, and greater partnerships with the community as well. Thanks very much.
Thank you. And don't forget the demonstration.